So good afternoon, everyone. First of all, I would like to thank the organizing committee for giving me the opportunity to present part of my PhD work here today. My name is Rita Domingues, and I am a third-year PhD student working at Acinetobacter Lab in Portugal. And today I'm going to talk about a heat-induced genome reduction strategy for Acinetobacter baumannii-infecting phages. So carbapenem-resistant Acinetobacter baumannii is the major cause of nosocomial infections worldwide. These strains are highly resistant to last-line antibiotics, causing very high mortality rates. So novel therapies are required to control these infections. And of course, phage therapy is one of them, and we've talked about many advantages of phage therapy already, but there are also some concerns. One of them being that most phages have many genes with unknown function. And here in the slide, you can see the genome of a small phage, a podophage with around 40 kb of genome size. And in white, you can see some genes with unknown function. And this leads to uncertainty in the application of phage therapy in human patients. So the goal of this work was to, first of all, optimize a heat-based technique to reduce the genome of an baumannii-infecting phage, then assess the fitness of the heat-treated phages and comparing them to the wild-type phage, and overall just contribute to the safety of phages for clinical use. So let me introduce you to the phage that I mainly work with, which is the 3043 phage. It's a podophage with around 42 kb genome size. And I am using this phage because it is highly studied in our group. And so we performed the heat treatment assays on this phage. And the image that you see displayed in the slide represents one cycle of heat treatment. So basically, we subject the phage through an incubation for 60 minutes at a high temperature. In our case, in the first cycles, we used 55 degrees. And later on in the final cycles, we increased to 65 degrees. And then the surviving phages are produced in the host strain, and then they are subject to the heat once again. And so we performed this for 20 cycles, and afterwards we sequenced the mutants through whole genome sequencing. And by whole genome comparison with the wild type phage, we could see that our mutant phages had deletions of around 2.2 kb, especially in the early genome of the phage and always encoding hypothetical proteins. Also, as you can see displayed in black in the slide, there are 4 deletion regions that were set between homologous repeated regions of 6 to 12 base pairs. And in red, you can see the spots where we found SNPs in 2 genes involved in host recognition that we could find. So then we decided to characterize our mutants to understand if they were as fit as our wild type phage. So we started with a one-step growth curve to understand if the infection cycle of our mutant would be the same as the wild type phage. And as you can see, there is no difference in the infection cycle of both phages, meaning that there is no apparent fitness cost to our mutant phage when comparing to the wild type. So we then performed some adsorption assays to understand if our heat-treated mutant would adsorb to our host cells quicker or if it would take longer than our wild-type phage. But as you can see, no difference in the adsorption time to the host between both phages. So then we performed more assays, one of them being the bacterial growth inhibition assay. And so we performed this assay for 24 hours, and let me guide you through the results. So the line with the circles represents the control line, which is basically our host cells without any treatment of phages. And the other 2 lines, which are pretty mixed together, are the lines that represent the treatment of our host cells with both phages. And as you can see, no difference in the bacterial growth inhibition by both phages. So finally, we did a temperature resistance assay where we incubated both phages for 60 minutes at different temperatures, and we used a range from 40 till 80 degrees, and we always started with a very high titer, 10 to the 10th. And so once again, let me guide you through the results. So here in this line with the circles is the wild type phage. And as you can see, when we increase the temperature, it tends to lose titer very quickly, specifically at temperature of 60, where it loses half of its original titer. And at 65, it's killed. While the heat-treated phage, which is here represented in this line with the squares, is able to maintain a very high titer, 10 to the 10th, from the 40 degrees until the 60 degrees mark. It loses some titer at 65, and at 70 is completely killed. So between 70 and 80 degrees, we have no phage. So this led us to conclude that our mutant phage is slightly more tolerant to higher temperatures than the wild type phage. So to conclude, the heat treatment assay is a very helpful strategy to select phages with smaller genomes. It selects phages with deletions of genes with unknown functions without compromising the fitness of the phage. And this is very important because it enhances the safety of phages for clinical use due to the lack of these genes with unknown function. And this strategy also facilitates further engineering of the phages, for example, to include payloads or reporter genes, therefore improving the efficacy of native phages in the control of baumannii infections. And with this, I finish my presentation. I would just like to acknowledge all of the funding entities, my whole lab team, specifically and most importantly, my supervisors, Professor Joana Azeredo and Dr. Hugo Oliveira, and of course, all of you for listening. And if you have any questions, I will be happy to answer.